Welcome back to part 3 of chapter 10. So we had just finished talking about oxygen debt and how we repay our cells, replenish the ATP, make more creatine phosphate, add the oxygen back to our hemoglobin and myoglobin, convert the lactic acid that we produced during our energetic period back into glucose that can be then stored as glycogen. And so now we're going to talk about muscle relaxation. So when muscles contract, they need to have a lot of calcium, and so our cells are flooded with calcium that binds to the troponin, that causes troponin and tropomyosin to move off the actin. But when that stimulus stops up here at the neuromuscular junction, and we stop having acetylcholine being released, then that's going to stop the action potential that's running along the sarcomere, and so we stop having calcium being released into the sarcoplasm. The calcium that's in the sarcoplasm is then going to be retransported back into the sarcomere. Now this takes ATP. So this is going to occur, so for muscles to relax, we need ATP, and for muscles to contract, we need ATP. We also need ATP for the myosin to release from actin. So without myosin binding to the actin, myosin is going to stay, or myosin binding to ATP, then myosin will stay bound to the actin and we'll have that continued contraction. So ATP is needed in both the release of the actin molecule and in the pumping of calcium back into the sarcoplasmic reticulum. So let's look at what that looks like, or let's go through each of the steps. So the first step is you're going to have an inhibitory neuron stimulating the excitatory neuron, basically telling it to stop sending signals. That leads to the excitatory neuron, the somatic motor neuron, no longer releasing acetylcholine. Any acetylcholine that's in the synaptic cleft then left over is going to be broken down by acetylcholine esterase, the enzyme that breaks down acetylcholine. ATP binds to myosin, allowing myosin to relax and release from actin. ATP also binds to the calcium pumps, pumping calcium against its concentration gradient from lower to higher concentration back into the sarcoplasmic reticulum. And this should actually be up here. I don't know. I kind of have that a little wrong. It should be 1, 2, 5, 3, 4. So 1, 2, get it. Apologies for that. All right, so now you've seen the muscle contraction. Let's talk a little bit about a disorder associated with muscle contraction called muscular dystrophy. So this muscle contraction that we've talked about is really looking at the myofilaments inside of a muscle fiber. Those myofilaments can move all they want if they're not actually attached to the muscle fiber, so to that endomysium of the muscle fiber, then it's not going to cause the muscle itself to contract. So dystrophin is a protein that links the actual sarcomere to the endomysium. And it's a genetic defect in the protein dystrophin that leads to muscular dystrophy, a group of diseases that causes muscle degeneration. So the most common form is called Duchenne muscular dystrophy. This is an X-linked recessive disorder. X-linked disorders, that's not a word. X-linked disorders are more often seen in males than in females. And this is because males only have one X chromosome, and since it's recessive, a female can carry the recessive disorder but still not have the disorder if she has a dominant chromosome as well. When you have this disorder, it causes muscles to not be able to contract, which causes muscles to shrink or atrophy. And eventually it leads to being unable to use those muscles, and it will lead to death of the individual, typically at a younger age. People can live to their 20s with muscular dystrophy, but most people don't live to their 30s or 40s. So let's look at different muscle contractions now. So there are 3 types of muscle contractions we can look at. We can look at concentric muscle contractions, where the muscle itself shortens. We can look at eccentric muscle contractions, where the muscle itself elongates. So that's what we typically see when we are contracting and relaxing our muscles. So concentric muscle shrinks, eccentric muscle lengthens, whereas in an isometric contraction, the muscle does not change size. It does contract, but it doesn't change in actual size. So there's no movement of the muscle. So think of with concentric or isotonic contractions, we have the muscle movement. So we're contracting, concentric, relaxing, or elongating, eccentric. These are isotonic movements. Isometric movement or isometric contraction, there is no change in the length of the muscle. Think of pushing a car. There's no way that you have enough tension. You cannot move the load because the load is so heavy that you don't have the muscle strength to move it. So pushing a car that's not rolling. That would be an example of an isometric contraction. And now let's look at the size of a contraction. So size of a contraction can really be dependent on the number of muscle fibers being stimulated. So a motor unit is one nerve fiber and all of the muscle fibers that innervate. So one nerve fiber can be composed of multiple motor neurons, and each neuron can have multiple end bulbs on its axon. Each end bulb will innervate one muscle fiber. And so when it stimulates, it can signal different muscle fibers to contract. And so when you have one nerve stimulating multiple muscle fibers, this is going to produce a weak contraction, depending. But if you have multiple motor units innervating the same muscle, causing them to contract, it's going to be a stronger contraction. So we have different sized motor units. We can have some motor units that only have a few muscle fibers that are stimulated per neuron, or we can have very large motor units where we have over, hundreds and hundreds, up to 1,000 neuron or up to 1,000 muscle fibers per neuron are stimulated. So small motor units, you're going to have fine motor control, whereas large motor units, you're going to have a lot of strength. So what is the ideal size of a sarcomere, or when a muscle contraction occurs, when is it going to be the strongest? The size of the sarcomere, if you have the sarcomere if it's contracted too much, it's not going to be a very strong muscle contraction because you can't work very well with a very, very short sarcomere. If you stretch it out too much, again, it's not going to be a very strong muscle contraction because those filaments, myofilaments, are too far apart. You're not going to get good contraction length. But if that sarcomere is between 80 and 120, that's going to give you the size of the sarcomere. That size is going to give you the best muscle contraction. So here, you're not going to be able to pull the myofibrils into that M line. Here, you're not going to have enough overlap to pull the myofibrils in. This one, they're already so close, it's not going to happen much. But this one is the perfect size for a nice, strong muscle contraction. So let's look at a muscle contraction now. So this is a myogram of a muscle twitch. So when you're looking at the x-axis, this is time in milliseconds. So these are 100 milliseconds. So all of these occur before 1 second occurs. Does that make sense? And then this is the y-axis is the tension that's formed. So when something stimulates our muscle fiber, a single muscle fiber, the stimulus occurs and it's going to take approximately a few milliseconds before the contraction occurs. So this is known as the latent period. This is when This is when acetylcholine receptors, This is when acetylcholine receptors open up or bind to acetylcholine and then allow sodium to rush in. Sodium rushes in, this stimulates calcium to flood the sarcoplasm, and then calcium binds to the troponin, tropomyosin move off of actin, and now we have that muscle contraction. And then from here, when we get to this top, we have sodium channels are closing. We have other channels opening up, potassium channels. Potassium rushes out, bringing back that regular resting potential of the muscle fiber. And we have this relaxation period. During relaxation period, calcium is booted out. The actin and the myosin go back to their resting lengths. Troponin and tropomyosin cover back over the actin molecule. Here we have TRPE. So in TRPE, we're increasing the stimulation frequency, and so we're still having complete relaxation occurring, but calcium channels are not all booting the calcium, all the calcium in the cytoplasm is not being booted out, so there's calcium left. So with the next muscle stimulus, more calcium is available, and so we can produce more cross bridges, causing stronger and stronger contractions. If we increase the stimulus even further, we end up having no relaxation period or less relaxation period, and so we end up with this wave summation where we're increasing the contraction until we eventually will hit this tetanus, where there's just a sustained muscle contraction. Any sustained muscle contraction can only last for a certain period of time until the muscle just fatigues and it just can no longer function. So this leads me to a couple of disorders. So again, this is known as tetanus, where the muscle is in a sustained contraction. This is a disorder called tetanus caused by bacteria known as Clostridium tetani. These are bacillus-shaped cells. They are streptobacilli cells. They're gram-positive for those of you who in microbiology. They are anaerobic bacteria that live in close relation to cows and other farm animals. So they're part of their normal flora. Clostridium tetani produce a toxin called the tetanus toxin. And this toxin, when it is released into the body, it moves to affects the nervous system, blocking inhibitory neurotransmitters. So remember, somatic motor neurons stimulate muscle contractions, but for them to stop, they need an inhibitory neurotransmitter that stimulates the somatic cell to stop releasing neurotransmitter. So this inhibits that somatic neurotransmitter, or it inhibits the inhibitory neuron from releasing neurotransmitter to stop the somatic motor neuron. And this will lead to muscle contraction that is sustained, known as tetanus, and that can be very, very dangerous, life-threatening. Another disorder caused by another bacterial species in the same genus, Clostridium botulinum, forms another neurotransmitter called botulism toxin. This toxin works on skeletal muscle by stopping it from contracting, whereas this one causes sustained contraction or spastic paralysis. This one here causes a sustained relaxation or no ability to contract. That's known as flaccid paralysis. In both cases, your muscles can't move, so they're paralyzed. With Clostridium botulinum, what happens is the excitatory somatic motor neuron normally releases its acetylcholine into that synaptic cleft. Botulinum toxin inhibits acetylcholine from being released, so the muscle can no longer contract. Some individuals will use, and I think I talked about this in chapter 6, this toxin, a modified form of this toxin, to actually diminish wrinkles in their face, which some people like. I'm not a big, I'm not someone that would do that, but you know, some people like it, so that's it is to each his own. That's all I can say about that. Another disorder associated with skeletal muscle and contractions is myasthenia gravis. So this is an autoimmune disorder where antibodies mimic, or I won't say mimic, they bind to acetylcholine receptors, so they have the same binding shape as the acetylcholine, but when they bind, they actually break down those receptors. They ruin or damage the receptors, and when they do so, they end up causing the muscle itself to no longer be able to bind acetylcholine, which leads to decreased muscle stimulation in those cells. One of the first signs of myasthenia gravis is eye drooping, which is also known as ptosis of the eye. And sometimes your face becomes more flaccid. So you'll see this in individuals with myasthenia gravis. I don't have a great picture, so I found this and I thought this would be good because it shows the acetylcholine receptors and acetylcholine bound. So what happens is antibodies will bind here and break down this receptor. So now we're going to go into how we classify muscles themselves. Skeletal muscle can be classified 2 different ways: by how fast the contraction occurs and by how the muscle produces its ATP. So we have fast and slow contractions, and then we have anaerobic means of producing ATP through that creatine phosphate, as well as through lactic acid fermentation. And then we have oxidative mechanisms, aerobic respiration. So in general, every muscle is going to have all 3 types of muscle fibers, but certain muscles are going to have more of one type of fiber than another. And so let's talk about each fiber type. And I always like to think of this as when you eat chicken, most people have a preferred type of meat. Some people like red meat, some people, no, that's so wrong. Yeah, no, it is chicken. It's not red though. It's dark meat and light meat. That's what they say. Do you like dark meat or do you like the white meat? Dark meat is like the legs, the thighs. The light meat is the wings and the breasts. So the dark meat is the meat that has the mitochondria for aerobic respiration. It has a dark red appearance because of all of the myoglobin with the oxygen. So your legs, which function in very long term, they need a lot of energy and they need to not be able to fatigue very fast, are going to be dark in color or have that red appearance because of the myoglobin. And so an example of one of these fibers would be the gastrocnemius. These are slow oxidative fibers because they contract slow, they relax slow, but they can function for long periods of time. So people who runners, so I'm thinking long distance runners, marathon runners, they're going to have a lot of slow oxidative fibers built up in their legs. And that's what I was talking about there. They still have the fast oxidative and fast glycolytic, but they have more of those slow oxidative fibers in those legs. Whereas fast oxidative fibers, they're going to still have a reddish color, but not as red because they do use mitochondria to produce energy, but they don't produce as much energy. They contract and relax very rapidly. They still use aerobic respiration, but they just don't use it very often because they don't contract as often. So an example of this muscle would be the tibialis anterior. And so now what I always like to say, okay, think about these muscles. You're using your tibialis anterior when you are on your heels. So stand up and put your heels on the ground and lift up your toes and walk around on your heels and see how that feels. You can do that for a shorter period of time, typically, than you can walk on your toes. So now I want you to step on your toes and walk on your toes. You could probably walk on your toes for, a few minutes, depending on your balance and everything. But most people can walk on their toes for a good amount of time, whereas you can't walk on your heels near as long because your tibialis muscle, tibialis anterior, is not as powerful, or is not, no, I won't say that, does not have, or it fatigues faster. That's what I want to say. It's very powerful, but it fatigues really fast, whereas your gastrocnemius is not as powerful, but it doesn't fatigue either. So it's slower contractions, but it'll last for a long period, if that makes sense to you. And then we have those fast glycolytic fibers. So think of your arm muscles. Arm muscles tend to be very, very strong, but they function for shorter periods of time. We don't tend to hold like very heavy things in our arms for a long period of time, whereas we're holding our body up on our legs all the time. And so fast glycolytic fibers are found again throughout all of our muscles, but we see these in certain muscles more often than others. And one example is in the biceps. So fast glycolytic fibers use anaerobic mechanisms to provide ATP. Because of this, they don't have the myoglobin. They don't need that oxygen. It's anaerobic. And so they're white. And this is where like your breast and your wings, so the wings or the arms of the chicken would be found. In us, our arms have more fast glycolytic fibers than they do slow oxidative or fast oxidative fibers. Very powerful, very fast, but very short bursts of energy. And so think about people who lift really heavy weights. They might lift 400 pounds over their head and then they have to drop it. They're done. Whereas you can push 400 pounds with your legs 3 or 4 times, and you can still do other things with your legs. Your legs have a lot more ability to function for long periods of time. They don't fatigue, but your arms will fatigue much faster in general. And this is just looking at a bodybuilder from your textbook, okay? All right, so the last thing we're going to look at, actually, I'm going to stop this lecture and we'll get into the last lecture for this last part so that I don't overwhelm you. So I'm going to stop it here, and I'll see you in just a few minutes. Bye.